He was running a trail he had run 100 times before. The path was familiar, the air was warm, and the morning felt ordinary in every way. Then his foot struck something in the ground, and within seconds, the air around him turned black. Not dark, black. A living, moving, furious black cloud that rose from the earth like smoke from a fire and came straight for him. 400 Africanized honeybees hit his body in under 2 minutes. He ran. He screamed. He kept running, and by the time he collapsed at the edge of the trail, his body had already begun a war with itself that would last for days. If you want to understand what venom does to a human being at that scale, what it means to survive something that has killed full-grown horses and cattle and healthy adults who never made it to the hospital, then stay with this. Because what happened inside his body is one of the most dramatic biological events a living person can experience and walk away from, and the fact that he walked away at all is something worth understanding in full. Before we go any further, if this is the kind of story that pulls you in, the kind that sits at the edge of what the human body can endure and what the natural world is capable of delivering, then make sure you subscribe right now. There is so much more where this came from, and you will not want to miss what is coming next. Now, let us go back to the beginning. Not just the moment he stepped on that nest, but back to the insect itself. Because to understand what 400 stings does to a person, you first have to understand what a single sting does, and why the Africanized honeybee delivers something so much more consequential than its European cousin. The Africanized honeybee is not a naturally occurring species in the way we typically think about wild animals evolving in isolation over millions of years. It is, in a very real sense, a human accident. In 1956, a Brazilian geneticist named Warwick Estevam Kerr brought 26 queen bees from southern Africa to Brazil. The goal was reasonable enough. African honeybees were known to be more productive in tropical climates. The idea was to crossbreed them with European honeybees already established in South America, creating a hybrid that would thrive in the tropics and produce more honey. What happened instead was something no one fully anticipated. 26 swarms escaped quarantine. That was all it took. The hybrid bees that resulted from those escaped queens were extraordinary survivors. They adapted to their new environment faster than almost anyone predicted. They spread north through South America, through Central America, through Mexico, crossing into the United States near Hidalgo, Texas in 1990. They moved at a pace of roughly 300 miles per year. They colonized deserts and jungles and suburban neighborhoods. They nested in the ground, in walls, in hollow trees, in water meter boxes, in overturned flower pots, in gaps in concrete. They were relentlessly adaptable, and they were, compared to the European honeybee, most people know from gardens and honey farms, in a completely different category when it came to defensive behavior. Here is the key difference, and it is a profound one. A European honeybee colony will typically send out 20 to 30 guard bees when disturbed. They will pursue a threat for a relatively short distance, perhaps 100 feet or so, and then they will return to the hive. An Africanized colony will mobilize hundreds to thousands of bees in response to a disturbance. They will pursue a threat for a quarter mile, sometimes more. They have been documented chasing horses and livestock into water. They have chased people into buildings and remained outside, circling for hours. They respond to vibration. They respond to the carbon dioxide in-breath. They respond to dark colors. They respond to movement. And once the alarm pheromone is released, once that first bee stings and releases that sharp, banana-like scent into the air, the response from the rest of the colony is cascading and almost impossible to stop. That is what this man ran into on the trail that morning. He was a runner in his late 30s, fit, healthy, no known allergies to bee venom. He had run this trail dozens of times. The nest was in the ground, likely established in a rodent burrow, as Africanized bees frequently colonize. His foot came down close enough to the entrance to send shockwaves through the earth. Within seconds, the first guard bees were on him. Within 30 seconds, hundreds more. He did exactly what survival guides tell you to do. He ran. He did not swat at them, which would have only worsened the alarm pheromone saturation around him. He pulled his shirt over his face to protect his airways. He ran in a straight line as fast as he could. He ran for approximately a quarter mile before the density of bees around him began to thin. He did not stop running for another 100 yards after that. By the time he finally stopped, he estimated later that the stinging had lasted between 3 and 4 minutes. He pulled more than 40 stingers from his face alone. The total count across his body, documented later at the hospital, was somewhere between 380 and 430 stings. The uncertainty in the number reflects the fact that by the time medical staff were able to do a proper count, significant swelling had already obscured many of the sting sites. Now, let us talk about what was actually happening inside his body during those minutes, and in the hours that followed. Because the story of what bee venom does at that concentration is a story of chemistry and biology and the human immune system operating under conditions it was never designed to handle. A single bee sting delivers roughly 50 micrograms of venom. The venom is a complex cocktail. The primary active component is melittin, a peptide that makes up about 50% of the dry weight of honeybee venom. Melittin is a membrane-disrupting molecule. It inserts itself into cell membranes and causes them to break apart. This is what creates the immediate burning sensation. It is not just irritation. It is actual cellular destruction at the site of the sting. Melittin triggers the release of inflammatory mediators, causes mast cells to rupture, and initiates a cascade of responses that draws blood and immune cells to the area. The second major component is phospholipase A2, an enzyme that degrades the phospholipid membrane of cells. It works alongside melittin to amplify cellular damage. It is also one of the primary allergens in bee venom, meaning it is a significant driver of severe allergic reactions. There is also hyaluronidase, which breaks down hyaluronic acid in connective tissue. This functions essentially as a spreading factor, allowing the other components of the venom to penetrate more deeply into tissue and spread more widely from the sting site. Apamin is a neurotoxin present in smaller amounts that crosses the blood-brain barrier and affects the central nervous system. Histamine is present in the venom itself, adding to the local inflammatory response independent of the body's own histamine release. In a single sting, these components cause local pain, redness, swelling, and heat. The reaction is typically self-limiting and resolves in a few hours to a few days. The immune system mops it up. It is unpleasant, but manageable. At 400 stings, none of this is manageable. The total venom load delivered to this man's body was somewhere around 20 milligrams. That is a significant toxic dose, even setting aside any allergic component. At concentrations like this, bee venom stops being a local irritant and becomes a systemic toxin. The distinction matters enormously. Local reactions stay where they start. Systemic reactions travel. Within the first 15 minutes after the attack, he was already experiencing what he described as a sensation of the world narrowing. Not quite tunnel vision, more like a dimming at the edges. His heart was hammering. His skin felt simultaneously burning and numb in different areas. He was nauseated. He was still on his feet, still moving toward the trailhead, driven by the pure biological imperative of survival. But the signals his body was sending him were shifting rapidly from alarm to something deeper and more serious. His wife found him at the edge of the parking lot. She described him later as gray-faced and shaking, with his face swollen to the point where she initially did not recognize him from a distance. She called emergency services immediately and kept him awake and talking while they waited. By the time paramedics arrived, approximately 25 minutes after the attack, his blood pressure had dropped significantly. His heart rate was elevated and irregular. He was vomiting. The swelling across his face and neck had progressed to the point where the paramedics' first and most urgent concern was his airway. One of the most dangerous consequences of mass envenomation or severe allergic reaction to bee stings is angioedema, swelling of the deep layers of skin and the tissues of the throat and airway. If the airway closes, everything else becomes irrelevant. He received epinephrine in the field. He received antihistamines. He was intubated as a precaution, meaning a tube was placed into his airway to ensure he could continue to breathe, regardless of how much further the swelling progressed. He was transported to a trauma center by helicopter. What was happening inside him during that transport is a story of biology under siege. The mass release of histamine from thousands of ruptured mast cells had caused widespread vasodilation. Blood vessels across his body had dilated simultaneously, dropping his blood pressure dramatically. This is the mechanism behind anaphylactic shock, and even without a classic allergic response, the sheer volume of venom was capable of driving this process through direct chemical action rather than through immune hypersensitivity. His blood was being flooded with vasoactive compounds faster than his body could compensate. His kidneys were in immediate danger. The kidneys filter blood. When blood pressure drops, Kidney perfusion drops. When kidney perfusion drops, the delicate cells of the renal tubules begin to die. But there was an additional threat to his kidneys beyond the blood pressure problem. Mass envenomation causes hemolysis, the destruction of red blood cells. Melittin attacks red blood cell membranes directly. When red blood cells are destroyed in large numbers, they release their contents into the bloodstream, including hemoglobin. Free hemoglobin in the bloodstream is toxic to the kidneys. It is filtered out but clogs the tubules and causes acute tubular necrosis, a form of sudden kidney failure. The breakdown products of muscle also flood the bloodstream as venom-damaged muscle cells release myoglobin, another protein that is deeply toxic to renal tissue. This combined process is called rhabdomyolysis, combined with hemolysis-induced nephrotoxicity, and it is one of the leading causes of death in mass B envenomation cases that are not immediately fatal from cardiovascular collapse. His liver was also under chemical attack. The enzymes released into the bloodstream from damaged cells across his body would begin to accumulate: liver enzymes, cardiac enzymes, muscle enzymes. The blood work being drawn and analyzed in the trauma center was telling a story in numbers, and the story was alarming. His heart. This is where the narrative becomes particularly dramatic, because cardiac complications from mass B envenomation are among the least intuitive but most serious consequences of this kind of attack. B venom contains compounds that act directly on cardiac tissue. Melittin itself is cardiotoxic. It disrupts the electrical conducting system of the heart. It causes arrhythmias. It can cause heart block, where the electrical signals that coordinate the chambers of the heart are interrupted or slowed. In severe cases, it has caused ventricular fibrillation, the chaotic and ineffective quivering of the heart's main pumping chambers that, without immediate defibrillation, is fatal within minutes. His cardiac monitoring in the first hours at the trauma center showed ST segment changes on the electrocardiogram. ST segment changes are among the findings that cardiologists look at when they are worried about myocardial infarction, a heart attack. The heart tissue was under direct chemical stress from the venom and indirect stress from the hemodynamic instability caused by the systemic reaction. He had not had a classic heart attack in the way a blocked coronary artery causes one, but the damage to cardiac cells was real and measurable. His inflammatory markers were extraordinary: C-reactive protein, interleukin-6, tumor necrosis factor alpha. All of the alarm signals of a body-wide inflammatory response were spiking. His white blood cell count was dramatically elevated. His immune system was not just responding to an allergen, it was responding to catastrophic tissue damage across multiple organ systems simultaneously. The coagulation system was under stress. Disseminated intravascular coagulation, sometimes abbreviated DIC, is a condition where the clotting cascade is activated so widely and simultaneously across the body that the clotting factors are essentially consumed. The body tries to clot everywhere at once and runs out of the materials it needs to clot anywhere. The result is paradoxical bleeding even as clots form in small vessels throughout organs. It is one of the most dangerous and difficult conditions in all of emergency medicine. In his case, early signs of coagulopathy were present, meaning the timing and extent of clot formation were abnormal, though he did not progress to full DIC. The intensive care team managing him was essentially fighting on 6 fronts at once, supporting his blood pressure with vasopressors and fluids, protecting his airway, supporting his kidney function, which had already begun to show signs of acute injury, with careful fluid management and monitoring for the need for dialysis, managing his cardiac rhythm, treating the venom-induced inflammation, and managing the pain, which was, throughout all of this, extraordinary. He would later describe the pain during the first day as unlike anything he had a reference point for. Not the sharp, specific pain of an injury. Something more total, like a deep, penetrating burning that seemed to come from inside his tissues rather than the surface. The sting sites themselves were raised and hot and individually painful. But the systemic pain, the muscle aches and the headache and the sensation of pressure behind his eyes and the cramping across his abdomen, that was something different. That was the body communicating at every level simultaneously that something had gone very wrong. He spent 4 days in intensive care. On the second day, his kidney function test showed acute kidney injury. His creatinine was elevated. His urine output dropped, and the urine that he produced was dark, a sign of the hemoglobin and myoglobin that had been filtered through his kidneys from the mass destruction of cells. The nephrology team was consulted, and the possibility of dialysis was seriously discussed. Aggressive intravenous fluids were used to flush the kidneys and maintain urine output. By the end of the second day, his urine output had improved. By the third day, his creatinine was beginning to trend downward. His kidneys had been injured, but they had not failed beyond recovery. His cardiac markers remained slightly elevated through the second day before beginning to normalize. He was monitored continuously for arrhythmias and was placed on medications to support and stabilize his cardiac rhythm. No significant sustained arrhythmia occurred after the first 12 hours, though several transient episodes of abnormal rhythm were detected and managed. The swelling across his face and neck began to recede slowly through the second and third days. This is a process that feels agonizingly slow to the person experiencing it. The swelling from bee venom at this concentration is not superficial. It extends into the deep tissues. His eyes were essentially swollen shut for more than 36 hours. The swelling of his lips and tongue was so significant that communication was limited to written notes and head movements for much of the first 2 days. His skin across the areas of highest sting density, his face, neck, arms, and the back of his legs where he had been most exposed during the run, showed widespread pustule formation as the local venom reaction in hundreds of individual sites progressed through its natural course. Each sting site goes through the same process. Initial pain and burning from melatonin and phospholipase activity. Histamine release causing redness and swelling. Immune cell recruitment causing the site to become a small focus of inflammation. In a person with a single sting, each of these sites would be a minor inconvenience. In him, there were hundreds of them, and they were all progressing simultaneously, covering significant portions of his body with a landscape of inflammation that had to be managed topically in addition to everything else. By the third day in intensive care, his vital signs had stabilized. He was taken off vasopressor support. His blood pressure was holding on its own. His cardiac monitoring remained on as a precaution, but showed sinus rhythm. The intubation had been removed, as his airway swelling receded enough for the team to be confident he could maintain his airway independently. He described the first moment of breathing through his own airway again, without the tube, as one of the most relief-laden experiences of his life. He was transferred out of intensive care on the 4th day and spent 3 additional days in a regular hospital room. The concerns at that stage were continued monitoring of his kidney function, management of the lingering pain and inflammation across his skin, assessment of his liver enzymes, which had been elevated and were slowly normalizing, and psychological support, because surviving something like this is not only a physical process. We will come back to the psychological dimension, because it is often the part of survival stories that gets the least attention and deserves far more. But first, let us think about why some people die from mass bee envenomation and others survive. Because the number 400 stings is often cited as somewhere near a threshold, and the truth is more complicated than a single number. The lethal dose of bee venom for a healthy adult human is typically estimated at around 500 to 1,000 stings. This is a rough statistical figure derived from documented fatalities and experimental data in animal models. It accounts for the venom load from roughly 25 to 50 milligrams of bee venom, but this number is enormously variable. A child has a lower body mass, and therefore a proportionally higher venom concentration per kilogram of body weight from the same number of stings. An elderly person with pre-existing cardiovascular or renal disease has far less physiological reserve to survive the assault. A person with a known or unknown allergy to bee venom can die from a single sting through anaphylaxis, a mechanism that has nothing to do with total venom load and everything to do with immune hypersensitivity. This man was fortunate in several specific ways. He was fit, with strong cardiovascular reserve. He had no known allergies to bee venom, and his reaction, while severe, was driven by toxic volume rather than immune hypersensitivity, meaning the most immediately life-threatening mechanism of anaphylaxis was not the primary driver of his crisis. He reached a high-level trauma center quickly. He was young enough that his organs had significant reserve capacity to sustain the insult and begin recovery, and he made the right decisions in the first minutes of the attack. That last point matters more than people realize. The instinct when something stings you is to stop and brush it away, to swat at it, to try to see what is attacking you. In an Africanized bee attack, any of these behaviors is potentially fatal. Stopping allows the swarm to concentrate on you. Swatting releases more alarm pheromone and signals to the colony that you are a significant threat, requiring more defensive response. The carbon dioxide from your exhaled breath acts as an additional attractant. The only correct response is to run in a straight line, to cover your face, particularly your nose and mouth, and to keep running until you are well clear of the swarm, ideally indoors, where windows and doors can separate you from the bees. He did all of this correctly, under conditions of extreme physical and psychological stress, with hundreds of stings already landing on him. That behavioral response, those 30 seconds of correct decision-making, likely saved his life as much as anything the medical team did afterward. Let us go deeper now into the longer recovery, because leaving the hospital after 7 days was not the end of the story. It was closer to the beginning of a second chapter. In the weeks following discharge, he experienced a range of symptoms that are well documented in survivors of mass envenomation, but are rarely discussed in the media coverage that typically focuses on the dramatic acute event. His kidneys, while they had recovered enough to avoid dialysis, showed continued subtle dysfunction for more than 3 months. His creatinine levels remained mildly elevated compared to his pre-attack baseline. Regular monitoring was required. The nephrology team was cautiously optimistic, but could not make any promises about long-term renal function for the first several months. His cardiac function was monitored with periodic echocardiograms, imaging tests that assess the structure and function of the heart's chambers. There was a small area of reduced wall motion detected in the first echocardiogram performed after his discharge, suggesting an area of myocardium that had been stressed or mildly damaged during the event. By the 3-month echocardiogram, this finding had largely resolved, which was genuinely good news. But the cardiologists were explicit with him. He had sustained real chemical stress to his cardiac tissue. The long-term implications were uncertain enough that he would need continued monitoring. His skin took weeks to fully heal. The pustule formation and crusting across hundreds of sting sites resolved slowly. Some of the areas of highest sting density developed what are called hyperpigmented scars, areas of darker skin that mark the location of significant tissue damage. Many of these faded over months. Some did not. He described looking at his arms in the mirror during recovery and feeling a kind of bewilderment at the geography of the damage mapped out across his own skin. There was something else that developed in the weeks after discharge that his medical team had anticipated, but that he had not fully prepared for. Hyperacusis to the sound of buzzing insects, not a dramatic phobia in the clinical sense immediately, but a profound and involuntary startle response to any buzzing sound. The sound of a lawnmower, the sound of a fly near his face, the sound of a bee in a garden, any of these would trigger an immediate and overwhelming physical response. Heart rate spiking, adrenaline flooding, the sensation of wanting to run. He had developed a classically conditioned fear response, the same mechanism Pavlov described in his dogs, but experienced in a human nervous system that had been through a genuinely life-threatening event. This is not weakness. This is neuroscience. When the brain experiences an extreme threat, particularly one that arrives so suddenly and so overwhelmingly, the amygdala, the part of the brain responsible for processing fear and triggering survival responses, lays down extraordinarily strong associative memories. The sound and visual sensation of swarming insects became associated with mortal danger in a single catastrophic learning event. The brain does not reason through this association. It does not pause to evaluate whether the single bee near the garden flowers is actually dangerous. It fires the same alarm response because, from an evolutionary perspective, firing too many false alarms is a much safer error than missing a real threat. Post-traumatic stress disorder was formally diagnosed approximately 6 weeks after his discharge. The nightmares began first. Vivid sensory dreams of the attack, the specific quality of the air going dark around him, the sound, the sensation of hundreds of simultaneous impacts. He would wake from these dreams in full sympathetic activation, heart racing, sweating, disoriented. Sleep disruption became a significant problem and contributed to the exhaustion and emotional dysregulation that characterized the first 2 months of recovery. He had intrusive waking memories as well, what clinicians call flashbacks, where sensory input in the present moment would trigger a partial re-experiencing of the trauma. Walking near any kind of ground vegetation would sometimes do it. A particular quality of warm air, the smell of certain plants. The nervous system had essentially written the context of the attack in exquisite detail and encoded it as a survival signal. The work of recovery from the psychological dimension was in some ways longer and harder than the physical recovery. He described it later as having to learn to teach his brain that it was allowed to stand down, that the threat was over, that the alarm could be quieted. Trauma-focused cognitive behavioral therapy and prolonged exposure therapy were both used as part of his treatment. Progress was real but non-linear. There were setbacks, weeks of improvement followed by a particularly bad night, or a particularly difficult encounter with an ordinary bee that undid what felt like progress. Approximately 18 months after the attack, he described himself as largely recovered from the psychological effects. He was running trails again, though different trails than before. He carried an epinephrine auto-injector. He wore light-colored clothing. He had learned everything there was to know about identifying and avoiding Africanized bee nests. The knowledge gave him a sense of agency in an experience that had been defined entirely by its sudden and total removal of his control. He said in an interview that the most important thing the experience had taught him was not about bees. It was about how thin the line is between an ordinary morning and a catastrophic one. He had crossed that line in less than a second, between one footfall and the next. And the lesson was not to live in fear of that possibility. The lesson was to recognize it and still choose to go outside, still choose the trail, still choose to be in the world where extraordinary things, both terrible and beautiful, happen without warning. Let us step back now and think about the Africanized honeybee itself, not as a villain in this story, but as an animal. Because these insects did not arrive at their current place in the ecosystems they now inhabit through malice. They arrived through adaptation, through the relentless biological imperative to survive and reproduce that drives every living thing. The African honeybee, from which the Africanized hybrid inherits its defensive behavior, evolved in an environment that was genuinely hostile in ways European landscapes were not. Sub-Saharan Africa has a far richer predator guild than Europe. Honey badgers, raytails among the most ferocious small mammals on Earth, raid bee colonies regularly. Bee-eater birds, primate raiders including chimpanzees and humans who have been harvesting honey from wild colonies for hundreds of thousands of years. The response of the African honeybee to threats evolved in direct proportion to the severity and persistence of those threats. Extreme defensive response, rapid mobilization, long pursuit, high sting threshold, all of these are solutions to an ecological problem. The bees that survived in Africa were the ones that defended most aggressively. The genes encoding that behavior were naturally selected for generation after generation. When those genes arrived in the Americas, the threat landscape was different. The predators were less experienced with bee colonies. The human populations encountering them had no cultural knowledge of the specific risk. The bees spread into environments from tropical rainforest to semi-arid desert with remarkable adaptability, and the defensive behavior that made perfect sense in a Kenyan savanna became a significant and sometimes fatal risk in a Texas suburb or a Brazilian farming community. There are documented cases of Africanized bee attacks killing dogs, goats, horses, cattle. There are cases of people dying from fewer than 200 stings when pre-existing health conditions reduced their resilience. There are cases of elderly individuals alone on properties where colonies had established themselves in walls or outbuildings who were discovered after fatal attacks with no one nearby to help them. The risk is real, and it is not distributed equally across the population. Beekeepers working with European colonies in areas where Africanized bees are established must deal with the progressive Africanization of their hives as Africanized drones mate with European queens, and the colony's defensive behavior shifts over generations. Managing this requires careful queen replacement programs and increased protective protocols. The honey industry in affected regions has had to adapt continuously. What is sometimes underappreciated is that Africanized bees are genuinely excellent honey producers. They forage more widely, work longer hours, and can establish viable colonies in a wider range of conditions than European bees. The agricultural value that was originally envisioned when they were brought to Brazil is real. The challenge has always been managing the defensive component, separating the productivity from the danger, a challenge that bee researchers continue to work on. There is also the story of what these bees do as pollinators. Across the regions they have colonized, Africanized bees serve as significant pollinators for both wild plant communities and agricultural crops. Their ecological role is not simply as a hazard. It is as a participant in the systems of reproduction and seed dispersal that maintain the plant communities on which everything else depends. The complexity of their ecological position reflects a broader truth about introduced species generally. They are not good or bad in any absolute sense. They are organisms responding to conditions, occupying space, performing functions, causing disruptions, all simultaneously. Now let us think about what this man's experience tells us about the relationship between humans and the natural world at its most unmediated and most intense. We live, most of us, at an extraordinary remove from the kind of contact with nature that his experience represents. The trail he was running on was a recreational amenity, a managed piece of landscape designed to give people access to green space while maintaining a degree of comfort and safety. And yet, underneath the surface of that managed landscape, a colony of Africanized bees had established itself in the earth. The natural world was there all along, doing what it does, completely indifferent to the recreational amenity that had been laid across the surface of its territory. There is something clarifying about a story like this, not because it argues that nature is dangerous and should be avoided, a counsel that would impoverish human life enormously, but because it reminds us of something we are very good at forgetting. We are animals. We move through an environment populated by other organisms that have their own imperatives, their own responses to threat, their own long evolutionary histories that have nothing to do with our comfort or our safety. Most of the time, those imperatives and ours coexist without incident. We walk past a wasp nest and the wasps assess us as a non-threat and return to their work. We disturb a spider and it retreats. The natural world is in most circumstances astonishingly tolerant of our presence. But the tolerance has limits, and the limits are not always visible. A ground nest is invisible. The decision by 400 bees to defend it is instantaneous and total. The gap between ordinary morning and extraordinary crisis is exactly as thin as one footfall. Across the American Southwest, in Florida, in Texas, in New Mexico, in Arizona, in California, Africanized bees are now established parts of the local ecology. They are not going away. Eradication is not feasible at this scale. The appropriate response from a human behavioral perspective is knowledge, understanding how to identify the signs of a colony nearby, the unusually intense activity around a particular spot on the ground, or a crack in a wall, or a gap in a fence post. Understanding what to do if disturbed, the absolute importance of running, of not stopping, of covering the face, of seeking enclosed shelter. Understanding the importance of not attempting to remove a colony without professional assistance. These things matter. They are literally life and death practical knowledge for people living in affected regions. There are pest management professionals who specialize in Africanized bee removal. The work requires full protective gear, careful assessment of the colony's location and size, and techniques that minimize the release of alarm pheromone during removal operations. It is skilled work, and it is important work. The increasing establishment of Africanized bees across the southern United States has generated an entire professional ecosystem around their management. Let us come back, one more time, to him. To the man on the trail, to the specifics of what his body did and what his body needed and what it means that he is alive. He was stung 400 times. He ran a quarter of a mile with bees actively stinging him. He reached his car, barely, and reached a parking lot where his wife found him. He was helicoptered to a trauma center. He spent 4 days in intensive care and 3 more in a standard hospital room. He spent months in a recovery process that involved his kidneys, his heart, his skin, his sleep, and his psychological relationship with the world. And then he went back out. Not to the same trail, but to trails. He went back to running in wild places. He went back to the morning air and the sound of birds and the sensation of moving through landscape. He went back because the alternative, to let that morning on the trail define and limit the rest of his life, was something he was not willing to accept. There is a kind of courage in that return that is worth naming clearly. It is not the reckless courage of someone who has not processed what happened to them or who is performing bravery for an audience. It is the considered courage of someone who has sat with the full weight of what occurred, who has done the work of healing physically and psychologically, who understands the risk clearly now in a way he did not before, and who has decided that the life he wants to live includes being outside in the world. He carries epinephrine, he wears light colors, he makes noise on the trail, he knows what to look for, he runs with his eyes on the ground ahead of him in a way he never did before. He has become, in a meaningful sense, a more careful inhabitant of the landscape he loves. And that, perhaps, is one of the truest things that extreme encounters with the natural world can do for the people who survive them. They can strip away the casual assumption that the world is safe and replace it with something better, not with fear, with attention, with a quality of presence and awareness that most of us who have never had to run for our lives from a swarm of 400 bees simply do not carry. The world is extraordinary, and it is indifferent, and it is alive in every direction. The trail is worth running. The morning is worth entering. And the bees in the ground beneath your feet are doing exactly what their ancestors evolved to do over millions of years in the heat of the African savanna, which is to survive, with everything they have, against whatever threatens them. That understanding does not make the encounter